Good day to you, Ma'am Grace. My report is all about proteomics and its current trends. Most of us already know of what is the basic definitions of proteomics. Yes, proteomics is a study of the entire set of proteins produced by a cell, tissues, or organisms. But there's more behind that complex field in cellular biology, and now let's dive into it. Proteomics involves the large-scale analysis of proteins, their structures, functions, interactions, modifications, and abundance within a biological system. Proteomics play a crucial role in advancing biomedical research, drug discovery, understanding disease mechanisms, and developing diagnostic tools and personalized medicine. Proteomics is the study of the entire set of proteins known as the proteome within a biological system. It involves the comprehensive analysis of protein expression, structure, functions, interactions, and modification in a specific context such as cell, tissues, or organisms. Proteomics aims to understand the complex network of protein and their role in various biological processes, including cellular signaling, metabolism, and disease mechanism. Proteomics utilizes a combination of experimental and computational techniques to investigate proteins at a large scale. These techniques include mass spectrometry, protein separation methods such as gel electrophoresis, protein-protein interaction studies, and bioinformatics analysis. The main objectives of proteomics are protein identification. Protein identification, one of the primary objectives of proteomics is to identify the catalog all the proteins present in a biological sample. This involves the use of various techniques such as mass spectrometry to measure the mass and charge of proteins and compare them to known protein sequence in databases. By identifying proteins, researchers can gain insight into the composition and complexity of the proteome. So, the protein structure and functions. Understanding the structure and functions of proteins is a crucial objective of proteomics. Proteomics techniques such as X-ray crystallography, nuclear magnetic resonance, or NMR, and cryo-electron microscopy cryo-EM, enabling the determination of proteins structures at high resolution. By studying protein structures, researchers can infer their functions, interactions, and mechanisms of action. This knowledge is essential for drug discovery, personal medicine, and understanding disease pathways. And the last is post-translational modification or PTM. PTM is modifications which are chemical modifications that occur on proteins after translation. So PTM play a critical role in a protein's functions, localization, and regulation, such as mass spectrometry enabled the identification and characterizations of PTMs, such as phosphorylation, glycylation, and acylation, and ubiquitination. Understanding PTMs helps elucidate the rules in cellular processes, disease mechanisms, and biomarker discovery. Now, I will proceed on elaborating the current trends of proteomics. The one current trends of proteomics is biomarker discovery and disease diagnostic. So BDDD proteomics play a significant role in disease biomarker discovery for Alzheimer's disease or ED by providing insight into the protein expression patterns, post-translational modification or PTM, and protein-protein interactions associated with the disease. Here's how proteomics contributes to biomarker. biomarker discovery for Alzheimer's disease. So, for example of biomarker, identification of amyloid beta and tau proteins. So proteomics techniques have been discovered is instrumental in the identification and characterizations of amyloid beta and tau proteins, which are key pathological hallmarks of Alzheimer's disease. By using mass spectrometry and other proteomic approaches, researchers can study the proteolytic cleavage patterns, PTM, and aggregation states of AB and tau proteins. These findings contribute to a better understanding of the disease and the development of biomarkers related to AB and tau pathology. The second is a drug development. Drug development proteomics techniques help identify the validate potential drug targets by analyzing the proteome of diseased tissues or cells. By comparing the proteomes of healthy and diseased samples, researchers can identify proteins that are differentially expressed or post-translationally modified in the diseased state. These proteins can serve as potential targets for drug development. So, for example of drug development, in the development of cancer therapeutics, which is proteomics has been instrumental in identifying specific proteins that are overexpressed or aberrantly modified in cancer cells. So, for these instances, human epidermal growth factor receptor 2 or HER2 is a protein overexpressed in a certain breast cancer. Proteomic analysis have identified HER2 as a potential target for drugs like trastuzumab herpetin, which specifically targets HER2 positive breast cancer cells. So, overall, proteomics plays a crucial role in advancing our understanding of biological systems, disease mechanisms, and drug development. It has wide-ranging application in various fields, contributing to advancements in healthcare, agriculture, and other areas of scientific research. And that would be all. Thank you so much for listening, ma'am.